Hello, today we will be presenting about humanized mouse models with endogenously developed human natural killer cells for in vivo immunogenicity testing of HLA class 1 edited iPSC-derived cells. The presentation will be made by Daniela Rodriguez and Mariana Oloy. Our presentation consists of an introduction, materials and methods, results, and finally our discussion. Introduction. This article focuses on the use of human-induced pluripotent cells, also known as HIPSCs, for cell treatment. Clinical trials are using one of two, either fully compatible autologous cells or stocked iPSC-derived human leukocyte antigen, also known as HLA cells. An experimental approach is being considered of depleting the HLA expression on the donated iPSCs, which would lead to them being universally compatible. By doing this, the new cells can break down the barrier to transplantation, which usually is an HLA mismatch. Natural killer, also known as NK cells, are responsible for targeting cells with low HLA class 1. NK cells play an important role in this new approach. Lack of in vivo models containing NK human cells has been an obstacle for the study of these strategies. Therefore, MSTRG mice, which is an immunodeficient mouse strain with improved human hematopoietic cell reconstruction, had been previously used. Scientists treated the mice with exogenous factors to reconstitute the NK cells levels in humans. This study aims to establish the suitability of these humanized mice for the research of immunogenicity testing regarding NK cells. Additionally, the immunogenicity of HLA-edited HIPSC-derived cells will be investigated in the newly developed NSG-SGM3 strain. Materials and methods. Various steps had to be taken in order for this experiment to take place as human cells were used. These steps were ethics approval, human hematopoietic stem cells, human immune cell reconstruction in mice, cell preparation, cell infusion and survival analysis, and finally statistical analysis. For the ethics approval, it was approved by the ethics committees of Kyoto University and Kumamoto University. It's in compliance with the Declaration of Helsinki for experiments involving human samples, and animal experiments are approved by university committees and conducted according to university regulations on animal experimentation. For the human hematopoietic stem cells, CD34 plus HHSCs derived from cord blood obtained from different sources, the purification of HHSCs using a CD34 plus cell isolation kit, and finally the freezing of HHSCs in fetal bovine serum, also known as FBS, and dimethyl sulfoxide. The materials and methods used for the human immune cell reconstruction in mice was the use of MSTRG and NSG-SGM3 mice for human immune cell reconstitution, the intravenous injection of HHSCs into irradiated mice, the treatment with recombinant human IL-15 and IL-15 receptor alpha to support the development and maturation of human NK cells, monitoring of body weight, blood cell composition, and human cell reconstitution, and finally treatment with RHIL-15 and RHIL-15RA-FC to enhance human NK cell development. For cell preparation, the generation of HLA class 1 depleted iPSC-derived hematopoietic progenitor cells, also known as IHPCS, differentiations of HIPSCs into CD43 plus hematopoietic cells, culture conditions and medium composition during differentiation stages, the preparation of immortalized megakaryocyte cell lines, IMMKCLS, and HIPSCs-derived cytotoxic T cells. Finally, the culture of K562 cell line. The materials and methods for the cell infusion and survival analysis consisted of a treatment of IHPCs and IMMKCLS with interferon G prior to the infusion, the labeling of cells for identification, the infusion of labeled cells into mice via trail vein injection, the collection of lung and spleen samples at specified time points, and finally the processing of samples and analysis of cell populations using flow cytometry. Finally, for statistical analysis was performed using appropriate software and the evaluation of data for normality and variance to determine suitable statistical tests. Results.
Human MSTRE mice competently assess the NK cell immunogenicity of HLA class I edited HIPSC-derived cells. MSTRG mice were humanized by engrafting HHSC and treated with HIL-15, HIL-15R alpha to reconstitute high levels of NK cells. Reconstitution of human hematopoietic cells was higher in female mice compared to male mice. As we can see in figure A, irradiation was involved in this week. The predominant cell population in humanized mice was CD19 plus B cells, while CD3 plus T cells were negligible. Myeloid cells were reconstituted at a higher percentage in female mice, and the predominant subtype of NK cells was the CD15 times CD16 plus major subset. In figure B, we can locate the predominant subtype of NK cells that was CD15 times CD16 plus as the plots at the bottom. So in figure C and D, it's demonstrated that WT and HLA-KO IMMKCL along with K562 cells were co-infused into humanized mice to assess cell rejection. The presence of HLA-KO IMMKCLS and K562 cells was inversely correlated with the NK cell level in the lung and the spleen, indicating NK cell dependent rejection. Co-infusion of WT, HLA-KO, and HLA-C retaining IHPC was performed and their levels were analyzed. The ratio of HLA-KO iPSC derived IHPC relative to WT IHPC was inversely correlated with the human NK cell level, indicating rejection by NK cells. HLA-C retaining IHPC showed tolerance to NK cells in this model. So the results demonstrate the accuracy of human MSTRG mice for monitoring immunogenicity. In the results for human NSGS mice show high levels of human chimerism and support human circulating NK cell development with exogenous HIL-15, HIL-15R alpha. The optimization of the regimen was performed in NSGS mice, which are genetically modified NSG mice expressing human SCFF, GM-CSF, and IL-3. The HHSC graft was treated with anti-HCD3 antibody to eliminate T cells and prevention of GVHD. 3 different combinations were compared to minimize strain on NSGS mice, considering irradiation, HHSC quantity and body weight, WBC, RBC, platelets, hemoglobin, and hematocrit measurements. The transplantation of 1/105 HHSC with 1G preconditioning was selected for stable reconstitution with less severe anemia and thrombocytopenia. Mice were treated with or without HIL-15, HIL-15R alpha to compare different levels of human NK cell reconstitution. The treatment with HIL-15 and HIL-15R alpha did not significantly affect body weight, blood cells level, but result in significantly increased spleen size in humanized mice. The comparison of spleen size between humanized and non-humanized mice show a significant increase in spleen size and in the humanized mice. As you can see in figure B, this one is bigger than the NSGS. Primaries in human immune cell reconstitution was constantly high, with slightly higher levels following HIL-15, HIL-15R alpha treatment. The CD19 plus B cells were the dominant population among HCD45 plus cells, followed by CD33 plus myeloid cells. The CD3 plus T cells and NKp46 plus NK cells numbers were low, but HIL-15 and HIL-15R alpha treatment increased NK cell numbers and slightly increased T cell numbers. The results demonstrate that HIL-15, HIL-15R alpha treatment can enhance the reconstitution of human NK cells with minimal effects on other cell types, providing a stable in vivo model with a high number of endogenously developed human NK cells. So the results for human NSGS mice reject HLA-KO cells depending on the reconstitution of human NK cells. For the results, the human NSGS mice were intraperitoneally injected with or without HIL-15, HIL-15R alpha. The functional capacity of reconstituted human NK cells to reject HLA-KO HIPSC derived cells was excellent. Figure B shows that HLA-KO IMMKCL and their WT counterparts were co-infused in a 1:1 ratio and labeled with fluorescent tracking probes. The rejection of HLA-KO cells in the lung was assessed 1 day, shown in the figure D1, and 3 days in the figure D3, later by flow cytometry. Rejection of HLA-KO cells correlated with the logarithmic level of reconstituted human NK cells in the lung, and a similar trend was observed in the spleen. Discussion. The article aimed to evaluate the suitability of the indigenous human NK cell reconstitution strategy to model missing self-immunity against HLA class I edited HIPSC-derived cells. Immunodeficient mouse strains with transgenic expression of human cytokines enhanced the reconstitution of human hematopoietic cells upon HHSC transplantation. Human NK cell reconstitution was observed in MSTRG mice treated with HIL-15, HIL-15R alpha, where MSTRG mice did not show this reconstitution. Similar observations were made with NSGS mice. The rejection of HLA-KO cells correlated with the level of human NK cells reconstitution in the lungs and spleen of human NSGS mice. Other mouse strains expressing human IL-2 or IL-15 transgenes may have similar utility without the need for administering exogenous cytokines. So in this article, it showed that in previous studies in vivo immunity using human peripheral blood mononuclear cells and NK cell lines in immunodeficient mice has shown transient reconstitution and limited reflection of indigenous developed NK cell immune response. The functionality of human NK cells to reject HLA class I downregulated cells that had only been confirmed against highly immunogenic malignant cells and not for non-tumor cells, including HLA-KO, HIPSC-derived cells. So the female MSTRG mice show higher human hematopoietic cells reconstitution than male mice. and NSGS mice exhibited higher reconstitution overall. Male NSGS mice might also be useful in vivo models. The in vivo rejection capacity correlated with the level of NK cell reconstitution as a percentage or absolute number in the lung, while spleen samples show more variable results. The humanized mice may suffer from deteriorating conditions such as anemia, thrombocytopenia, enlarged spleen size, and wasting, indicating the need for better control of GVHD and systemic effects of cytokines. The rejection of HLA-KO, IHBC, and tolerance of HLA-C retaining IHBC in human NK mouse models demonstrate the role of HLA-C as an inhibitory ligand against human NK cells. The human NK mouse models can contribute to assessing the in vivo immunogenicity of diverse HLA-KO cells against endogenously developed human NK cells. The models can also be used to assess the immunogenicity of HLA haplotype homozygous cells and the preclinical testing of HIPSC-derived T cells as off-the-shelf products. The rejection of HLA-KO IMMKCL suggests the possibility of preparing HLA class 1 derived platelets while implementing fail-safe measures against the contaminating cells expressing oncogenic transgenes. The NK cell-dependent rejection response was observed in the lungs and spleens of different cell types, indicating that human NK mouse models are suitable for the infusion of various cells and tissue transplantation studies. And finally, HLA editing HIPSC-derived cells requires solid preclinical testing, and this study provides an in vivo evaluation of NK cell rejection tolerance for such cells, contributing to the clinical application and